Hello, Arun.
Yes, sir. Your
Yes.
slide accessible.
Okay, thank you. So I will start right away. A very good evening. So today we are going to have ninth live session for the interaction for solving the question by chemistry. So I will be taking few questions and solving them. So first question is that which one of the following statement is true with reference to enzyme? It says epenzyme, halenzyme, epenzyme is equal to halenzyme plus coenzyme. Halenzyme is equal to epenzyme plus coenzyme. Coenzyme is equal to epenzyme plus halenzyme. Halenzyme is equal to epenzyme minus coenzyme. What's the correct answer here? Any guess or any answer? So answer B is correct. Halenzyme. Answer B is right. Yes. Halenzyme is equal to epenzyme plus coenzyme. Epenzymes are non-dialigible protein macromolecules that can be destroyed by heat, whereas coenzymes are molecules of a small size that cannot be destroyed by heat. Since coenzymes are not proteins, together they form a halenzyme. Okay, so halenzyme is equal to epenzyme plus coenzyme. So what are the examples of another question associated here? What are the examples of this epenzyme and halenzyme? So the DNA polymerase and RNA polymerase are two haloenzymes which require magnesium ions to bind to become active and initiate polymerization. It consists of the protein part of enzyme. Its example like catalytic component of DNA polymerase enzymes are called as apoenzyme. Okay, so overall the haloenzymes is equal to apoenzyme plus coenzyme is the correct answer. Let me see who is joining. So next question we have, you are working at a company evaluating compounds with the potential to become a new medications. Okay, you evaluate several inhibitor of an enzyme you are studying and get the data below corresponding to inhibition constant Ki, which would be the most promising lead compound. So the basically it is asking if you have a Ki value for different inhibitor for the enzyme. So which will be the best inhibitor for the enzyme? That compound only will be the lead compound. So here Ki value is given. So what do you think, Arun and Sakshi?
Option C was the correct answer.
Which one?
C. Option C.
Okay, why do you think option C is correct? Option C is correct. Okay, why do you think it is option C is correct? Any reason behind that? Because in C only it is a lower value, right? So the inhibition constant K is the common case of competitive inhibition can be obtained by simple comparison of progress curve in presence and in absence of inhibitor. The difference between times taken for concentration of substrate to fall to the same value is used to obtain Ki. So the lower the Ki of the particular drug at particular receptor, the stronger its binding affinity for that receptor. The lower the value of the drug, the receptor drug, the stronger is the binding affinity. So this is because the lower Ki means that the drug can occupy 50% of this receptor even when the drug is present in a lower concentration. So what whenever a drug is needed, if the concentration is very low, required to inhibit the 50% of the activity. So that is a very potent drug. Okay, so in this cases, the Ki is 1.5 into the 10 to the power minus 8 molar is the lower value. Okay, so option C is correct. So value with the C compound will be the lead compound. Okay.
Yes, sir.
Okay. So next question we have. Okay, a prosthetic group of a protein is a non-protein structure, that is a ligand of a protein, a part of the secondary structure of a protein, a substrate of a protein, permanently associated with the protein.
Option D, permanently associated.
Option D, permanently associated. Yeah, right. So prosthetic group assists cellular function by. Okay. So is a type of helper molecule which is a non-proteinaceous compound that help enzyme to perform function. Coenzyme is a specific kind of cofactor molecule which is organic molecule that help enzyme to catalyze chemical reaction. Okay. So permanently associated with the protein. Another question we have, enzyme that differ in amino acid sequence but catalyze the same reaction are cofactor, coenzyme, apoenzyme, isoenzyme. Enzyme.
Isoenzyme.
Okay, isoenzyme, right. So option D is correct. So isoenzymes also are alternative forms of the same enzyme activity that exist in different proportion in different tissues. Isoenzymes differ in amino acid sequence composition sequence composition and the multimeric quaternary structure mostly but not always. They have the similar structures. And cofactor is a non-protein chemical compound or a metallic ion that is required for enzymes roles as a catalyst. So cofactor can be considered helper molecule that assist in biochemical transformation. Okay. So here is the simple depiction of the cofactors, apoenzymes, coenzyme. So you see this is the cofactor, this is the coenzyme and apoenzyme, totally is haloenzyme. And coenzyme is defined as organic molecule that bind to the active site of the certain enzyme to assist the catalysis of reaction. More especially, coenzymes can function as an intermediate carrier of electron during reaction or transfer between enzyme and as a functional group. Next question we have, carbon monoxide is toxic to human because what is toxic to humans? Hello. Yeah, can anyone of you tell why this coenzyme are toxic to humans? Why this CO is toxic to humans? Carbon monoxide binds to the
Option B, sir.
option
B.
B.
Sir, option B.
It binds to heme portion of hemoglobin and cause heme to unbind from hemoglobin. No.
No, sir. It binds to the globin protein of hemoglobin and prevents the binding of O2.
Okay, it binds to the globin protein of hemoglobin and prevent the binding of O2. No, it do not bind to the globin protein of the hemoglobin. Uh, Carbon monoxide binds to the iron. Okay, and it prevents the binding of O2 to the iron. Option C is correct. Okay, so it prevents the binding this binding of O2 once. I'll show you next slide how this O2 binds to the heme. Then you'll understand in iron. Fine.
Yes, sir.
So next we have which one of the following statements is true? Competitive inhibition is reversible and can be overcome by increasing the concentration of substrate. Competitive inhibition is reversible and cannot be overcome by increasing the concentration of substrate. Non-competitive inhibition is normally irreversible but can be overcome by increasing the concentration of substrate. Competitive inhibition is reversible and cannot be overcome by increasing the concentration of substrate. So, which one statement is true?
Option A.
Option A. Yeah, right. Option A is correct. So, competitive inhibition is reversible and can be overcome by increasing the concentration of substrate. So, competitive inhibition is not the irreversible process. Non-competitive inhibition normally reversible but can be overcome by increasing the. But this is also not right, and this is also not right. So, option A is correct. Next, we have a question. This salivary amylase is an enzyme in human that breaks down starch. The optimum pH for this reaction is 6.7. The rate of this reaction would not be affected by. What do you think? What do you guys think for this?
Option C, sir.
Option C is enzyme concentration.
Yes.
Salivary amylase is an enzyme in human system. The optimum pH for this 6.7. The rate of reaction will not be affected. If you decrease or decrease or increase the enzyme concentration, it will not the rate of the reaction will not be affected. Yes, it will affect it, right? If you change the enzyme concentration, it will be affected.
Substrate concentration.
Substrate concentration, if you change the substrate concentration, also it will be affected. So if you maintain the pH of the reaction at 6.7, then the rate of the reaction will not be affected.
But sir, it's optimum pH of reactions. So
6.7,
how?
right? So it is asking if you maintain the pH of the reaction, then the rate of the reaction will not be affected. So option A is correct. The styling enzyme found in saliva that break down the insoluble starch found in food into small soluble sugar fragment. This is another name of amylase found in saliva. Another question we have again: when oxygen binds to heme containing protein, the two open coordination bonds of Fe2 plus are occupied by one oxygen atom, one amino acid side chain, one O2 molecule, and one amino acid acid atom, one O2 molecule, and one heme atom, two O atom. What's the correct statement here?
C.
One O2 molecule and one heme atom. Okay, no, this not the correct. Option B is correct. One auto molecule and one amino acid atom. I'll show you how this oxygen bind to the
Histidine, I think.
yes. So this is a heme group. Uh, heme has a porphyrin, right? This group, complete group, is called heme group. That has a iron as a metal center. So once it is bounded to the this proteins, then it will coordinate with the histidine. One end will be coordinated with the histidine. Then if the oxygen is present, then another coordination uh, site will be coordinated by oxygen atoms. So option B is correct. Small right now. So here the binding of this O2 to the heme. So option B is correct. Is clear? Okay, next question we have. One of the enzyme involved in glycolysis, aldolase, required zinc 2 plus for catalysis. Under conditions of zinc deficiency, when the enzyme may lack zinc, it would be referred to as. It will be called apenzyme, coenzyme, haloenzyme, prosthetic group. So if that enzyme requires zinc 2 plus and there is a deficiency of zinc 2 plus, so then that enzyme will be referred as.
Option A was the correct answer.
A is the correct answer. Yes. Apoenzyme or apoprotein is an enzymatic inactive protein part of an enzyme which requires a cofactor for its activity. Apart from catalytic RNA, most of the enzymes are proteins. Not all of enzymes require cofactor. Enzymes that do not require any cofactor are known as simple enzymes, like trypsin. So it will be a apoproteins. Another question we have apoenzymes. Okay, another question we have in the following diagram of the first step in the reaction catalyzed by the protease chymotrypsin. The process of general base catalysis illustrated by the number and the process of catalytic covalent catalysis illustrated by number. So here it is. This is chymotrypsin, and this is one number, two number, and three number is given here. They are asking the general base catalysis illustrated by which number. So what should be filled here?
Option B.
Option B, 1 and 3. Yeah, 1 and 3. Correct. Option B is correct. Myoglobin and the subunit of hemoglobin have a very different primary and tertiary structure, very similar primary and tertiary structure, very similar primary structure but different tertiary structure, very similar tertiary structure but different primary structure.
Option B is the correct answer.
Option
B.
very similar primary and tertiary structure.
Yes.
Option D is correct. So myoglobin and hemoglobin are globular heme proteins. When the myoglobin is where the former is a monomer and later is a heterotetramer. Despite the structural similarity of myoglobin to the alpha and beta subunit of hemoglobin, there is a functional difference between the two proteins owing to the quaternary structure of the hemoglobin. Another question we have: choose the incorrect statement about the active site of enzyme. The active site is a three-dimensional cleft. The active site takes up a large part of the total volume of enzyme. Substrate are bound to enzyme by multiple weak interactions. The specificity of binding depends on the precisely defined arrangement of the atom in an active site. Which option is correct here?
Option C. C was the incorrect option.
Substrate are bound to enzyme by multiple weak attraction.
Yes.
Option B is correct. So this option B is correct. So statement B is incorrect. So it is saying active site takes up large part of the total volume of enzyme. No, active site is just not a large part of the total enzyme activity. Right? It's a part, but it's not a large part.
Yes, sir. Yes.
So option B is correct. B is correct. So it is incorrect. Catalytic triad of the most serine proteases contain aspartic, histidine, and serine residue. Which of the following is true? The aspartic residue is used in acid catalyzed attack and the peptide bond. The histidine residue is responsible for substrate specificity of trypsin. The serine residue used as a nucleophilic attack on the peptide bond. Aspartic residue is responsible for the substrate specificity of the trypsin.
Option C.
Hmm?
C.
Option C. Okay, it is correct. Any other opinion on that? Yes, option C is correct. So here is the catalytic triad given. So this serine is attacked to the carbonyl of the amide bond. Another question we have: an enzyme that joins the end of two strands of nucleic acid is polymerase, ligase, synthetase, or helicase.
Ligase.
Hmm? Ligase. Okay.
Synthetase.
Option B is ligase correct. So in case of vitamin B6 disorder of amino acid transamination take place, which class of enzyme catalyzes this reaction? It is oxidoreductase, hydrolase, ligase, transferase. So which class of enzyme this catalyze the transamination? So it is a transferase.
Transferase.
Yeah, it is a transferase. Following stabilization of this transition state during lysozyme catalysis, the NAG4 product is released after attack of water molecule on oxonium ion, release of water molecule from the glycosidic bond, breakdown of a tetrahedral intermediate serine 195, a major conformation change in the enzyme. So when the NAG4 product is released, so this is a peptidoglycan breakdown of the peptidoglycan by lysozyme C. Yes, so option A is attack of water snake molecule on oxonium ion. So here's the mechanism of this. The lysozyme catalyzes the hydrolysis of a glycosidic bond between peptidoglycan. Okay, so it cleaves this bond, glycosidic bond, and here's the mechanism. So this is oxonium bond is formed, and then water molecule attack here. So option A is correct. Okay, so next question we have an allosteric inhibitor of an enzyme usually bind to the active sites, participate in feedback regulation, causes the enzyme to work faster, denature the enzyme.
Binds to the active site, sir.
Participate in feedback regulation, I think.
Yeah, participate in feedback regulation. Another question we have, optimum pH for the activity of lysozyme is 5.0. The active site of lysozyme C contains a glutamic acid residue, pK is 5.5. Aspartic acid residue, pK is 4.0. Which of the following statements is correct about the mechanism of lysozyme? C. The glutamic acid residue is in more polar environment than the aspartic acid. During the mechanism, an oxyanion transition state form. During the entire catalytic mechanism, the aspartic acid residue remains unprotonated. The glutamic acid residue acts as the general base catalyst. So just basis of pK and pH. Just guess. Find out the right answer here.
Option C, aspartic acid remained unprotonated
Why?
due to its lower pK value 4.0. So at the first he gets unprotonated, then glutamic acid. So
Okay. Yeah. Right. Yeah. Right. So here's the simple mechanism of this glycosidic bond cleavage. So these are the this is the aspartic acid. Aspartic acid. This is glutamic acid and aspartic acid. So in the overall mechanism, this aspartic acid remains deprotonated, unprotonated, and this glutamic 35 catalyze the reaction. And the question we have is acute pancreatitis is characterized by lack of synthesis of zymogen enzyme, continuous release of zymogen enzyme into gut, premature activation of zymogen enzymes, inactivation of zymogen enzyme. Option C, premature activation of zymogen enzyme. Okay. Out of which sort of following is a substrate specific enzyme? The hexokinase, thiokinase, the decocarboxylase lactase.
Hexokinase.
Hexokinase, not to hexokinase is the initial enzyme for glycolysis, catalyzing the phosphorylation of glucose by ATP to glucose 6 phosphate. So it is not specific toward the substrate. Thiokinase is the ligase that synthesizes coenzyme thioestates. It is also not specific to a substrate. Decarboxylase is also not specific. So lactase is very specific toward lactose. So option D is correct. Is that clear? Hello.
Yes, sir.
So option D is correct here. Next question we have, which of the following coenzyme take part in the hydrogen transfer reaction in electron transport chain? So it's tetrahydrofolate, methylcobalamin, coenzyme Q, biotin. So in the hydrogen transfer reaction in electron transport chain, which coenzyme is
C. Coenzyme Q.
coenzyme Q? Right. So here's the electron transport chain, and this is the Q, which takes part in hydrogen transfer. This is the Q coenzyme. Another question we have: Group one coenzyme participate in which of the following reactions? Oxidation reduction, transamination, phosphorylation, dehydrogenation.
Phosphorylation.
Phosphorylation. Okay. Oxidation reduction. So group one coenzyme participate in oxidation reduction. Next question we have: Thymine deficiency decrease cellular metabolism because thymine is a coenzyme for pyruvate dehydrogenase and alpha ketoglutamate dehydrogenase. Sorry. So thymine deficiency decreases cellular metabolism because thymine is a coenzyme for pyruvate dehydrogenase and alpha ketoglutarate dehydrogenase. Activity of ketolase is inhibited. It is required for process transamination. It is a cofactor in oxidative reduction. What is the correct answer here?
D was the correct answer. It was a cofactor in oxidative reduction, sir.
Option A is correct.
Option A.
Thymine is a coenzyme for pyruvate dehydrogenase and alpha ketoglutarate dehydrogenase. Option A is correct here. So there's a thymine phosphate is a coenzyme of many enzymes, most of which occur in prokaryotes. Pyruvate dehydrogenase and 2-oxoglutarate dehydrogenase complexes as well as in transketolase are the example of thymine-dependent enzyme present in eukaryotes, including humans. So degree of inhibition alpha by a competitive inhibitor is obtained from measurement of Vmax, measurement of Km, measurement of the y-intercept on this Lionel-Berg plot or crystallography studies. The degree of inhibition is
Option A was
measurement of Vmax.
yes.
Any other? Any other option? Any other opinion on this?
Km, measurement of Km.
Measurement of K. Yes, measurement of K. Option B. The degradation of RNA by pancreatic ribonuclease produces two nucleoside 2-phosphate, nucleoside 5' phosphates, oligonucleosides, one nucleotide 3' phosphate, and an oligonucleotide. So if this RNA is degraded by this RNase A, then what it will produce?
Option C.
Option C, oligonucleosides. Once the RNA is degraded by this RNase A, it will produce one nucleoside, 3' phosphate, and oligonucleotide. So option D is correct. So here's the this is one RNase A strand. If it is degraded by RNase A, then it will generate this 3' phosphate and an oligonucleotide. Another question, where if chymotrypsin molecule undergoes a serine 195 alpha mutation, then chymotrypsin will not bind to the substrate. Chymotrypsin will bind to substrate as well as cause cleavage. Chymotrypsin will bind the substrate but will not cause cleavage. No effect will be observed. So, what do you guys think about this? If the serine 195 is mutated with the alanine, then what will
Option
happen? Yes, yes, please.
Option C.
Why do you think option C? If alanine is muted, then still the substrate will bind.
Yes, sir.
It will bind. So means how the binding affected in chymotrypsin, right? It
Yes,
is
sir.
it is specific for hydrophobic amino acid, right? So substrate will bind, but it will not cause the cleavage because cleavage for the causing the cleavage. So there is 195 attack to the this amide bond of the substrate, right? Is
Yes,
that
sir.
is that correct?
Yeah.
Yeah. Option C is correct here. Okay. So phospholipids are molecules that contain long water soluble carbon chain, positively charged functional group, hydrophilic heads and hydrophobic tails, amino acids. So the answer is given here itself. Phospholipids are class of lipids whose molecule has a hydrophilic head containing a phosphate group and two hydrophobic tails. derived from fatty acid joined by an alcohol residue, usually a glycerol molecule. Okay,
Yes, sir.
so here this is, as you can see, this is hydrophobic head, and this is hydrophobic head and hydrophilic tail. Option C is correct. Okay. The glycerophospholipids are composed of two fatty acids attached to glycerol and one of the following head group: ethanolamine, choline, serine. So option one and second and third, first and third, one, second, third. So glycerophospholipids are major constituent of membrane and are responsible for membrane being a bilayer. These are more complex structure consisting of a molecule of glycerol to which are attached two fatty acids, a phosphate, and usually one other small molecules. So option D is correct here. Another question is, we are having the melting property of fatty acids and lipid barrier is due primarily to van der Waals forces, hydrogen bond, hydrophobic effect, electrostatic interactions. So melting properties of fatty acids is because of van der Waals forces. Another, cholesterol is essential for normal membrane function because it expands the thickness of a bilayer, keep membrane fluids, and catalyze lipid flip-flop in the bilayer, plug up the cardiac arteries. So why the cholesterol is important? Anybody? Keeps the membrane fluid. Cholesterol is waxy, fat-like substance that found in all the cells in your body. The body needs some cholesterol to make hormone vitamin D substance that help you digest food. So option B is correct. What are the membrane structures that function in active transport? Peripheral protein, carbohydrate, integral protein, or hydrophobic molecules? What are the membrane structures that function in active transport? So it is integral proteins which function in active transport. The membrane structure that function in active transport proteins are lipids. Protein function in active transport as pumps. These proteins bind to molecules and move them from an area of low concentration to an area of high concentration against the concentration gradient. So integral proteins which are involved here. All of the following are found in membrane except nucleic acid, phospholipid, glycoproteins, glycolipids. Yeah. What is the correct option here? So correct option is nucleic acid. Nucleic acid is not found in membrane. So which of the following is not an example of lipid found in lipid linked proteins? So these are the lipids, fatty acids. Which fatty acids is not found in lipid linked proteins? So it is this stearic acid.
Option A.
Option B. Option B is correct over here. And then is plasma membrane are made of protein, lipid, carbohydrate, protein, lipids, protein, carbohydrates. Which option is correct here?
Option C.
Options. Option.
C. C of the correct answer.
Plasma membranes are made of protein, lipid, and carbohydrates. So option A is correct. And the question: the simplest hydrophilic moiety present in the membrane lipid is the phosphate group, the hydroxyl group, the amino group, the group, and the glucose. So it is hydroxyl group. Very simplest hydrophilic moiety. The resting membrane potential is mainly determined by the chlorine ion gradient, sodium plus gradient, potassium plus gradient, calcium ion gradient. So it is determined by potassium and gradient. So what generates the resting membrane potential is the K plus that leaks from the inside of the cell to the outside while leak K plus channels and generate a negative charge on this inside of the membrane versus outside. So at least membrane is impermeable to Na plus as all of Na channels are closed. So option C is correct here. Which of the following considers the structural parent of this sphingolipids? This sphingosine, lecithin, sphingomyelin, or ceramide. So in this case, option D is correct. Ceramide is the parent of this. If you see the structure here, the ceramide, and from this sphingosine is made up from the ceramide. So To break down this bond, you'll get this sphingosine. So option D is correct. Which of the following induce conformational change in protein? It is a facilitated diffusion. Mm, Option C is correct here. Another is the most abundant membrane lipid in the biosphere: phospholipid, galactolipid, sphingolipid, or ether lipids. It will be galactolipids. These are the most abundant lipids in the biosphere. Receptor for steroid hormones usually resides in plasma membrane, cytoplasm, nuclear membrane, or nucleoplasm. So it is receptor for hormones, steroid hormones used to be in nucleoplasm. Okay, another question we have: the resting potential is dependent upon the electrochemical gradient of potassium ion because cell contain cell contains largely potassium. The permeability to sodium ions is very small except during action potentials. The permeability to the resting membrane to potassium is higher than for other ions. Of the other activity of sodium potassium ATPase. So I think which option is correct here? So option C is correct in this case. The permeability of the resting membrane to potassium is higher than other ions. So thanks a lot for this session. So today we have learned about till this lipids. So hope to see you next session next week. Thanks a lot for joining. Thank you everyone.